Thank you. I'm essentially going to put on the brakes here and begin by underscoring one point that Tom made. There's a couple of ways that I could underscore this point. I think what I'm going to say is that we shouldn't forget that humans are animals, and that as a consequence of our animal nature, if you will, is that we can be arrayed along a phylogenetic tree, and we're related to other living things in that way. And in line with the little bit of conversation that broke out at the end there that was very nice, the premise of this work, the kind of big picture lurking in the background, is that rats are not people, mice are not people, monkeys are not people, flies are not people, but we can learn from these various organisms and test the hypothesis that there may or may not be something in it for us. So I'm going to take you through a very rapid show, a walk through some different species. This is slime mold. I'm going to studiously avoid the fish and the bees and the ants. But this is a picture of slime mold. And in the absence of food, these guys secrete a signal, aggregate, create this structure that's called a fruiting body. And those that come together to form the fruiting body are essentially laying down their lives. They have no future. And spores are preserved, taken up into the top, and then eventually released. Those are the ones who go on. And there are people who have written about this as a form of altruism. These are harvester ants, and I'm not going to describe their biology, but you can see across the top row that there are differences in their size, and those differences correspond to the social role that they occupy. So in answer to one of the questions from up here earlier, if you were asking about ants, the question would have been, yeah, there are certainly characters that predict what they're going to be doing in their social role. And here's a variety of other animals somewhere in between, engaged in either parental or romantic acts. Okay? And here's where humans come in, and I'm choosing not to emphasize political or socioeconomic status type gradients here. These are just examples of social output in the form of a self-portrait by the artist Egan Schiele, who's demonstrating a sense of social isolation, frustration. In the case of the poet Yeats, who is one person, speaks to a whole world and describes an intimate interaction that we might all consider to be social. This is an example of, this is actually very interesting. I grabbed this off the web and then wound up calling the magazine to find out whether I could get a better image, because it's highly pixelated. It turns out that this, and they wouldn't send it to me, it turns out that this magazine is made for people who live in the Hamptons, and its primary function is to instruct women on essentially how to display themselves. This is a designer providing a display trait to various women in the Hamptons, and these sorts of things seep into culture. And this is my favorite. This is the John Coltrane Quartet. Some of you may know who this is. If you don't, I would suggest that you check him out. He's here to indicate that humans aggregate under the right conditions, like this or like a concert. And he's also here because that unit in particular was like a social world in its own right. These guys communicated with each other in an unparalleled way and in an unprecedented way, and as a consequence of their ability to improvise and spontaneously interact, were able to affect others, a highly social act. Okay, so what about flies? Not usually considered social organisms. I want to run through some examples of social behavior that come from my lab. You'll be hearing about others through the meeting. Ed Kravitz will be talking about aggression, and Marla will be talking about the foraging gene. And I'm just going to call attention to things done in my lab. I'm going to tell you about some work by Josh Krupp, who's a postdoc in the lab, who used an experimental design that I had used as a postdoc. What we're doing here is working with a wild-type strain and creating a comparison where the control is just wild-type flies kept in a light-dark cycle. And the question is, what happens when you house wild-type flies with a mutant fly? Does that affect the wild-type flies? So in the end, we're comparing wild-type to wild-type, and we're going to call that comparison wild-type to host. And this is a picture of gene expression. I realize it's not getting under the skin, but it is getting under the cuticle, and it probably represents a form of biological embedding. Okay? And what this shows in a very simple and straightforward way is that profiles of gene expression taken from animals with the same genetic background, these are inbred strains, subjected to differences in the way they're housed, will express their genes at different levels. The genes here describe a timekeeping mechanism. And over on your right, the DSAT1 gene is a gene that's important for metabolism of cuticular hydrocarbons. These are compounds that are used as pheromones, and they're involved in communication. And we also looked at patterns of these pheromones as they occur and found that the ability of the fly to communicate with other flies was probably impacted by its social environment, because the levels of hormones that we could measure on these flies, it's not hormones, sorry, pheromones on these flies, was dramatically affected by this simple manipulation. And so was mating. It turns out that flies maintained in a more diverse group mate more frequently. And that's plotted here in a very crude way over a time axis in a light-dark cycle. The stars there represent significant differences in the frequency of mating, but what you're not seeing is a difference in the temporal distribution of mating, but we see that too. That was shown by another postdoc in the lab, a guy named Jean-Christophe Billeter, who's now working in Groningen in the Netherlands. And he did an experiment where he was able to show that females taken from one wild-type strain called Cantonese, shown in black, mate with males from the same strain in this pattern. I'm just going to put it in this pattern over time. Females taken from another wild-type strain, another inbred strain called Oregon R, mate with their own type of males in a very different temporal pattern over time. So that's nice. That's what these strains do. We then placed Oregon R males with Cantonese females and got out the Cantonese pattern almost identically. And we placed Cantonese males with Oregon R females and got out the Oregon R pattern almost identically. And what that tells us is that the females are regulating the temporal expression of mating. Again, this is sort of a matter of the group dynamics. And the last example I'll give you is sort of a striking example because it goes into the next generation and it reveals what looks like an emergent property of mating in these flies. On the bottom here, again, you see males. There's going to be 6 males, either Cantonese in the shaded area or Oregon R in the white area. And what we're seeing here is that these Oregon R females will always, when they mate with Cantonese, there will always be hybrid offspring. It's got to be that way. They're the only choice for mating. When they mate with Oregon R males, they're going to put out males that are Oregon R in white. But what was remarkable here was that no matter what the mix of males was, if both types of males were available, the females would somehow put out a ratio of 3 hybrids to 1 Oregon R offspring, 75-25. You need a microphone.
We're recording.
Okay. Excuse me. Thanks. Is that better? Okay. So what I'm showing you here is that somehow the females are regulating variability in the next generation. And what's even more remarkable about this is not shown on the graph, and that is that there was not a single mating that gave us this pattern. This pattern emerged in a highly reproducible way by looking at the population level. It wasn't there in any individual case. And this is something that we're looking into now. I don't have an explanation for it, but it's an example of what happens. And what we'd like to know is, what are the flies doing? What are the group dynamics that explain this sort of thing? So I've sort of told you that locomotor activity is affected. This was my postdoctoral work that I referred to, and that the frequency and temporal pattern of mating is affected by the group, that levels of gene expression are affected, the levels of pheromone expression. I didn't show you a graph showing that numbers of progeny are affected, take my word for it. And I have shown you that genetic variability in the F1 is affected. So flies somehow figure out that the group makes a difference, and they make use of that. We wanted to explore that in a more rigorous way by looking at the dynamics. And so to do that, we got together with a good friend of mine, Michael Dickinson, who's over on your right. Michael is now at the University of Washington and was at Caltech at the time. He and an engineer named Pietro Perona designed a system for tracking flies called C-Tracks. And John Schneider, who's here, he does all of his own stunts, and I don't know where he is, but he's somewhere in the room. There he is in the back. John Schneider did the work that I'm going to be telling you about now. He went out to Michael's lab and learned how to use C-Tracks. And this is a brief example. I'll be showing you a couple of what an interaction between flies look like. I want to call your attention to the touching. I'll get back to that at the very end if there's time. But as you can see, we're able to look at and quantify or quantitate the interactions. And after doing this for quite a while and observing them on my desk and all over the place, we came up with various criteria to define an interaction. They are as follows: that the angle from the center of one fly to the center of another and then out the head of that fly should be 90 degrees or less, that there ought to be one body length between them, or 2 body lengths in total between the center of mass of those ellipses, and that this has to hold up for at least 1.5 seconds. And when we see that, we call that an interaction. And then we do, well, let me show you what this looks like. Sorry. Okay. The boxes are describing where interactions are occurring. They're depicted over here in the middle with the angle, the body distance, and the seconds given. And on your right is a kind of a history diagram that shows the same distribution of cue balls that are like flies over here with arrows connecting them. At the end, if we were to sit here and watch the whole thing, and I'd love to do it, but there isn't time, you would see a network diagram. This is just to give you an idea of what an experiment looks like. There are 12 flies in there. And what we do with them is very much like what Tom described. And I'm going to walk you through that very quickly. So we get a sequence of interactions and order them over time, and then we put them in a matrix. And in our matrix, we don't allow flies to interact with themselves. That's not part of the definition. But we can ask who's interacting with who and at what time. And we build up an idea about flies interacting in this way. What we do typically is we look for so there's 132 possible interactions, and we look for about 33 of these, or 25% of unique interactions, and say that density is what's going to work for us as a network. We've gone back and validated what we see there for other densities, and so I'll show you some of those data, but this is just to explain what we do. We can then get an idea of who's interacting with who, and that's what's shown here. The direction of an arrow shows who the interactor is and who's receiving the interaction, and this is what the network, what a network might look like with 12 flies and a bunch of interactions. And we're able to figure out the degree of each fly. And the degree just represents how many interactions are going out and coming into an individual. It's a sum of those. So you can see that this guy is sending one out, receiving one back from this individual, and receiving from one who has 7, so he has a total of 3. And we can do this for every fly, and then use the variance of each of those numbers as a metric that represents the network. So that's what we do. And we do this. These experiments go on for 30 minutes. And in that time, there will be thousands of interactions in some cases. And so to make use of all the data, we'll get 32 interactions and then drop the first one and slide down and compute a second one. And we iterate and use a sliding window or boxcar method in that way to build up a picture of what the network looks like overall. I hope that's reasonably clear. If it isn't, I can explain it later. And all of the data points then give us a mean, and that represents the network. So in some cases, we look at that individual snapshot, but in most cases, we look at the whole network. And the first question we need to establish is whether the networks we're seeing would arise just by chance. It could be that these things just occur randomly. So we can develop a random distribution called an Erdos-Redney distribution, statistical randomness, where we get a distribution of variances of 12 nodes interacting 33 times. And that gives us an idea about statistical randomness. We also have a more biological sense of randomness that involves controlling for what 12 flies do in the arena. Maybe they're bumping into each other. Maybe they're bouncing off the walls. How do we know that their interactions are intentional? And so we address those kinds of questions by taking trajectories from different flies of the same type from different experiments and putting them together and measuring a virtual network. And so now we have a virtual network control or an artificial network control and a random control. And we can ask whether a wild type, 20 wild type male networks is different from random, and in this case it is. It sits between the median of the artificial networks and the median of the random networks. And what we saw is that this was true for all of the wild type strains that we looked at and for both sexes. We looked at the sexes separately here. And we're now in a position to ask questions about what's going on in the network. Here's a simple question. Does an individual in the network know something about another individual's position in the network? And we can answer that question by looking at, we let our measurement go on through, say, 28 interactions, and we say how many flies has each fly interacted with at that point? And then we go ahead and ask, what's the chance that an incoming interaction will come to that individual after 28 interactions occur? And it turns out that the more popular an individual is, the more likely that more interactions will come in. So based on the way these talks are going, it's a lot more likely that Tom will be talking to a group of you after this session than I will, because he's among friends. We also want to have a way to control networks, to compare different networks between different strains. And so we looked at different network properties. Now, the property I'm going to focus on is one called betweenness centrality. And I'm going to try explaining betweenness centrality 3 ways and then move on, because I had a hard time explaining it to Marla last night. And so I want to make sure that people are with me if they want to be. The simple definition of betweenness centrality is a network, is a measure of the number of shortest paths through a node. That's the definition. And if you think about it and you live with it, it makes sense and it's all good. Another way of thinking about that is that betweenness centrality indicates the relative significance of an individual between other individuals. So they may have to go through that individual to get to them. And so an example that might work for you would relate to, say, my relationship to the president of my university. If I wanted to see the president of my university, I would have to go through my chairman's secretary, my chairman, the dean's secretary, the dean, the provost secretary, the provost, the president's secretary, and then the president. But if I were Allison Fleming, to pick on Allison Fleming, I might just pick up the phone, because I'm Allison Fleming. And if, on the other hand, I wanted to see my chairman, going through the secretary and measuring or the administrative assistant, and measuring her number would then become very high because she would be the one way to get there, and she would stand between us. And this is sort of indicated here on this on these two diagrams. The third way, you can see it. That high between us centrality is shown over there on your right, low is on your left, and I don't want to beat this up because it's eating time. We looked at between us centrality across a range of densities. That's the number of interactions that we're filling within the matrix. And what we saw is that at very low and very high densities, things kind of fell apart. And we think that that's because when the number of interactions was very low, there wasn't enough to measure, essentially. And when it was very high, the matrix was too clogged. And we're working on proving that. But what we see is a highly reproducible pattern indicating a strain-based difference between these two inbred wild-type strains on between the centrality. This is a difference in a group-level phenotype, if you will, and what it suggests is that there's some kind of genetic basis to it. The genomes of these two strains have been sequenced. They're essentially different genotypes, but at the very least, they're inbred differences, and what we're seeing here suggests that there are alleles within a population that will affect the way individuals interact, both within a population and between populations. And so we're now in a position to begin to look for that. Here's kind of a cool finding at the level of a fly group. When you look at between the centrality and you don't normalize to make the comparisons, you just compare the scores, what you can see is that it's a very stable event across all those iterations in the boxcar filter. And here's another measure that's even more stable, and the variability is so tight I had to throw it up anyway, even though I haven't presented it to you, because it just illustrates that these network properties are very stable. But when you look at the position of an individual within a network in iteration number 1, and you compare that to the position of that individual in the network at iteration 1,000, there's no meaningful correlation. It's a flat line. What that suggests, again, is that at the level of a group, there is a stable group phenotype, but that the individuals who are contributing to this group phenotype are not locked into a certain position or role, if you will. That's just something interesting to consider. We can talk about that if you'd like. The last point I want to make is that we did experiments to control for previous experience. And what that means is we looked at flies who were housed together before the experiment and compared them to flies who had been isolated as adults before the experiment and compared those to flies who had been drawn from different groups, housed together with other flies but strangers to one another. And what we saw there was essentially no difference. And that's also very interesting. It suggests that somehow we're tapping into a structure that manifests rapidly and that does not depend so much on prior experience. That doesn't mean that prior experience doesn't matter. And it doesn't mean that the quality of life or the nature of their interactions or what they're communicating about is the same in all cases. But what it says is that there's something highly embedded about the way these individuals interact with one another. And I want to stop on that note and vote on the little discussion that was going on at the end of the last presentation and say I don't think that it's going to be convergent. I think that there are going to be genes that will come out of an analysis like this that will eventually help us understand and predict mechanisms in other species. I would say, my collaborator Michael Dickinson might disagree, but I would say possibly with even with humans. And there's precedent for that in the fly literature. But I want to stop and give people a chance to breathe. If there are any questions, I'd be happy to hear it. Thank you. One more thing. I summed up, but I didn't show you one last movie. So if I have a second, I want to show you something. We discovered something while doing this that's really kind of amazing, and that is that when flies interact, they touch a lot. They touch a lot. And this is something that had been reported many, many years ago, and forgotten. People now are not aware of this. You're going to see this fly walking upside down on the glass ceiling. They like to do this. And these guys are going to touch. That's not much. This fly up here is doing something called grooming or preening. We don't really know what that means, but there are taste receptors on the legs. And so she may be sort of cleansing her palate, if you will. But check out this interaction. I would argue to Steve Sumi that my flies are as warm and caring about each other as your monkeys. And we see this is an anecdote, but we see a lot of this. It's not over. These are two females, right? Well, that's a good question, and I could give you my answer. They're using the middle legs, the hind legs. There you go. So now, that's it. Thank you very much.